A very warm greeting to all the viewers. Today we are going to discuss about the embryonic stem cells. Introduction. Embryonic stem cells are pluripotent stem cells derived from the inner cell mass of a blastocyst. Pluripotency refers to a stem cell that has the potential to differentiate into any of the three germ layers: endoderm, that is interior stomach lining, gastrointestinal tract, and the lungs; mesoderm, for example as seen in muscle, bone, blood, and urinogenital; or the third, ectoderm, that is found in epidermal tissues and nervous system. This session includes the information about stem cells derived from embryonic tissues. Stem cells are undifferentiated cells that can divide continuously to produce daughter cells. These daughter cells then undergo differentiation into particular cell types. Now, embryonic stem cells, also known as ESC, are an extraordinary class of stem cells that can proliferate indefinitely in culture into different cell types and tissues, as they possess very high developmental potential. This depends on the site at which they are introduced, and this exhibition of behavior is called pluripotency of the cell. The cells of the body, when grown in culture, generally maintain their original character. This is possible since each type of specialized cell has a memory of its developmental history. For instance, embryonic stem cells in liver adopt the character and behavior of normal liver cells. These stem cells have the remarkable potential to develop into many different cell types in the body during early life and growth. Moreover, in many tissues, they serve as a sort of internal repair system, dividing essentially without limit to replenish other cells as long as the person or animal is still alive. When a stem cell divides, each new cell has the potential either to remain a stem cell or become another type of cell with a more specialized function, such as muscle cell, a red blood cell, or a brain cell. Stem cells differ from other cell types. All stem cells, regardless of their sources, have three general properties. They are capable of dividing and renewing themselves for long periods. They are unspecialized. And lastly, they can give rise to specialized cell types. A starting population of stem cells that proliferates for many months in a laboratory can yield millions of cells. If the resulting cells continue to be unspecialized, like the parent stem cells, the cells are said to be capable of long-term self-renewal. Scientists are trying to understand two fundamental properties of stem cells that relate to their long-term self-renewal. Until recently, scientists primarily worked with two kinds of stem cells from animals and humans: embryonic stem cells and non-embryonic, that is somatic or adult stem cells. The functions and characteristics of these cells will be explained in this lecture. Scientists discovered ways to derive embryonic stem cells from early mouse embryo in 1981. The detailed study of the biology of mouse stem cell led to the discovery of a method to derive stem cells from human embryo and grow the cells in the laboratory in 1998. These cells are called human embryonic stem cells. The embryos used in these studies were created for reproductive purposes through in vitro fertilization procedures. When they were no longer needed for that purpose, they were donated for research with the informed consent of the donor. In 2006, researchers made another breakthrough by identifying conditions that would allow some specialized adult cells to be reprogrammed genetically to assume a stem cell-like state. This new type of stem cell are called induced pluripotent stem cells. Sources of embryonic stem cells. Embryonic stem cells as their name suggests, are derived from embryos. Most embryonic stem cells are derived from embryos that develop from eggs which has been fertilized in vitro in an in vitro fertilization clinic and then donated for research purposes with informed consent of the donors. They are not derived from eggs fertilized in a woman's body. Blastocysts stored in vitro fertilization clinics prove to be the potential source of embryonic stem cells. In normal development, the blastocyst would implant in the wall of the uterus to become the embryo and continue developing into a mature organism. Its outer cells would begin to form the placenta, and the inner cell mass would begin to differentiate into the progressively more specialized cell types of the body. When the blastocyst is used for stem cell research, scientists remove the inner cell mass and place these cells in a culture dish with a nutrient-rich liquid where they give rise to embryonic stem cells. Embryonic stem cells seem to be more flexible than adult stem cells. They are also generally easier to collect, purify and maintain in the laboratory than adult stem cells. Scientists can induce embryonic stem cells to replicate themselves in an undifferentiated state for very long periods of time before stimulating them to create specialized cells. This means that just a few embryonic stem cells can build a large bank of stem cells to be used in experiments. Embryonic stem cells would first need to be differentiated into specialized cell types if they are to be used for therapeutics. There are two sources of embryonic stem cells. One is in vitro fertilization, also known as IVF, and the other is nuclear transfer. The largest potential source of blastocysts from stem cell research is from in vitro fertilization clinics. This would facilitate the isolation of stem cells with specific genetic traits necessary for the study of particular diseases. The creation of stem cells specifically for research using in vitro fertilization is, however, ethically problematic for some people because it involves intentionally creating a blastocyst that will never develop into a human being. The second source, which is nuclear transfer, basically a process which is accomplished by inserting the nucleus of an adult differentiated cell which stimulates to form blastocyst from which embryonic stem cells can be generated. The stem cells that are created in this way are therefore copies or clones of the original adult cell because their nuclear DNA matches that of the adult cell. Scientists believe that if they are able to use nuclear transfer to derive human stem cells, it could allow them to study the development and progression of specific diseases by creating stem cells containing the genes responsible for certain disorders. In the future, scientists may also be able to create personalized stem cells that contain only the DNA of a specific patient. The embryonic stem cells created by nuclear transfer would be genetically matched to a person needing a transplant, making it far less likely that the patient's body would reject the new cells than it would be with the traditional tissues transplant procedures. Although using nuclear transfer to produce stem cells is not the same as reproductive cloning, some are concerned about the potential misapplication of the technique for reproductive cloning purposes. Other ethical considerations include egg donation, which requires informed consent and the possible destruction of blastocysts, culture and characterization of embryonic stem cells. Moving further, let's talk about how to grow these embryonic stem cells in laboratories. Well, growing cells in the laboratory is known as cell culture. Human embryonic stem cells are generated by transferring cells from a pre-implantation stage embryo into a plastic laboratory culture dish that contains a nutrient broth known as culture medium. The cells divide and spread over the surface of the dish. The inner surface of the culture dish is typically coated with mouse embryonic skin cells that have been treated so they will not divide. This coating layer of the cells is called a feeder layer. The mouse cells in the bottom of the culture disc provide the cells a sticky surface to which they can attach. Also, the feeder cells release nutrients into the culture medium. Researchers have devised ways to grow embryonic stem cells without mouse feeder cells. This is a significant scientific advance because of the risk that viruses or other macromolecules in the mouse cells may be transmitted to the human cells. The process of generating an embryonic stem cell line is somewhat inefficient, so lines are not produced each time cells from the pre-implantation stage embryo are placed into a culture dish. However, if the plated cells survive, divide and multiply enough to crowd the disc, they are removed gently and plated into the several fresh culture dishes. Embryonic stem cells that have proliferated in cell culture for 6 or more months without differentiating are pluripotent and appear genetically normal are referred to as an embryonic stem cell line. At any stage in the process, batches of cells can be frozen and shipped to other laboratories for further culture and experimentation. The process of replating or subculturing the cells is repeated many times and for many months. Each cycle of subculturing the cells is referred to as a passage. Once the cell line is established, the original cells yield millions of embryonic stem cells. Furthermore, it is also important for us to determine or identify whether the cells are embryonic stem cells or not, and do they exhibit their fundamental properties. So, to identify their fundamental properties, scientists at various points during the process of generating embryonic stem cell lines test the cells. This process is called characterization. Scientists who study human embryonic stem cell have not set any standard series of tests to determine the properties of the cells. However, there are certain common tests that are followed by all throughout the world, some of which includes inspection under microscope to check that the cells are healthy and undifferentiated by determining whether they can be regrown or subcultured after freezing, thawing or replating, and knowing the pluripotency of cell. Now, as long as the embryonic stem cells in culture are grown under appropriate conditions, they can remain undifferentiated or unspecialized. But if cells are allowed to clump together to form embryoid bodies, they begin to differentiate spontaneously. They can form muscle cells, nerve cells, and many other cell types. Although spontaneous differentiation is a good indicator of embryonic stem cells being healthy, the process is uncontrolled and therefore an inefficient strategy to produce cultures of specific cell types. So, to generate cultures of specific type of differentiated cells, for example, heart muscle cells, blood cells, or nerve cells, scientists try to control the differentiation of embryonic stem cells. They change the chemical composition of the culture medium, alter the surface of the culture dish, or modify the cells by inserting specific genes. Through years of experimentation, scientists have established some basic protocols for the directed differentiation of embryonic stem cells into some specific cell types. Significance and applications. Stem cells are important for living organisms for many reasons. In a 3 to 5 day old embryo called a blastocyst, the inner cells give rise to the entire body of the organism, including all of the many specialized cell types and organs such as the heart, lungs, skin, sperm, eggs, and other tissues. In some adult tissues such as bone marrow, muscle, and brain, discrete populations of adult stem cells generate replacement for cells that are lost through normal wear and tear, injury, or disease. Given their unique regenerative abilities, stem cells offer new potential for treating diseases such as diabetes and heart diseases. However, much work remains to be done in the laboratory and the clinic to understand how to use these cells for cell-based therapies to treat disease, which is also referred to as regenerative or reparative medicine. Laboratory studies of stem cells enable scientists to learn about the cells' essential properties and what makes them different from specialized cell types. Scientists are already using stem cells in the laboratory to screen new drugs and to develop model systems to study normal growth and identify the causes of birth defects. Research on stem cells continues to advance knowledge about how an organism develops from a single cell and how healthy cells replace damaged cells in adult organisms. Stem cell research is one of the most fascinating areas of contemporary biology, but as with many expanding fields of scientific inquiry, research on stem cells raises scientific questions as rapidly as it generates new discoveries. The specific factors and conditions that allow stem cells to remain unspecialized are of great interest to scientists. It has taken many years of trials and error to learn to derive and maintain stem cells in the laboratory without them spontaneously differentiating into specific stem cells. For example, it took 2 decades to learn how to grow human embryonic stem cells in the laboratory following the development of conditions for growing mouse stem cells. Likewise, scientists must first understand the signals that enable a non-embryonic or an adult stem cell population to proliferate and remain unspecialized. Stem cells have the fundamental property that does not have any tissue-specific structures and that allows it to perform specialized functions. However, unspecialized stem cells can give rise to specialized cells including heart muscle cells, blood cells, or nerve cells. When unspecialized stem cells give rise to specialized cells, the process is called differentiation. While differentiating, the cell usually goes through several stages, becoming more specialized at each step. Scientists are just beginning to understand the signals inside and outside the cell that trigger each step of the differentiation process. Scope and ethical concern. Stem cells offer opportunities for scientific advances that go far beyond regenerative medicine. They offer a window for addressing many of the biology's most fundamental questions. Watching embryonic stem cells give rise to specialized cells is like peeking into the earliest development of the many tissues and organs of the human body. Stem cell research may help clarify the role genes play in human development and how genetic mutations affect normal processes. They can be used to study how infectious agents invade and attack human cells, to investigate the genetic and environmental factors that are involved in cancer and other diseases, and to decipher what happens during aging. Stem cells may also revolutionize traditional chemical medicine because embryonic stem cells can continue to divide for longer period of time and produce a variety of cell types. They could provide a valuable source of human cells for testing drugs or measuring the effects of toxins on normal tissues without risking the health of a single human volunteer. In the future, Thousands of compounds could be quickly tested on a wide assortment of cell types derived from stem cells, making drug discovery more efficient and cost effective. Stem cells may also help scientists calculate the effects of toxic substances in drugs, food, and the environment. Nevertheless, scientists and society as a whole must consider the ethical implications of stem cell research. As discussed throughout this session, different ethical issues are raised by the wide range of stem cell research activities. The controversy over embryonic stem cell research touches on some of the same fundamental questions that society has grappled with in the debates over contraception, abortion, and in vitro fertilization. The questions at the center of the controversy concern the nature of early human life and the legal and moral status of the human embryo. Embryonic stem cell research often involves removing the inner cell mass from excess blastocysts that are unneeded by couples who have completed their fertility treatment. This prevents those blastocysts from continuing to develop. Although such blastocysts would likely be discarded and thus destroyed by the clinics in any case, some believe that this does not make it morally acceptable to use them for research or therapeutic purposes. They believe that the life of a human being begins at the moment of conception and that society undermines a commitment to human equality and to the protection of vulnerable individuals if blastocysts are used for such purposes. Some cultures and religious traditions oppose the use of human life as a means to some other end, no matter how noble that end might be. Other traditions support embryonic stem cell research because they believe that the embryo gains the moral status of a human being only after a few weeks or months of development. Many traditions emphasize obligations to heal the sick and ease suffering, goals for which embryonic stem cell research holds great potential and favor embryonic stem cell research for this reason. Summary: stem cell research is both an evolution and a revolution in modern biomedicine, especially in regenerative medicine. It can be regarded as a milestone on a progression of single findings and developments. Despite its revolutionary nature, the concept of using stem cells for therapeutic purposes has much in common with the now conventional principle of organ transplantation. Stem cells are undifferentiated cells that can be divided continuously to produce daughter cells. These daughter cells then undergo differentiation into particular cell types. Now, embryonic stem cells, also known as ESC, are an extraordinary class of stem cells that can proliferate indefinitely in culture as they possess very high developmental potential. The culture embryonic stem cells can develop into different cell types and tissues. This depends on the site at which they are introduced, and this is termed as pluripotency of the cell. Embryonic stem cells, as their name suggests, are derived from embryos. Most embryonic stem cells are derived from embryos that develop from eggs that have been fertilized in vitro. When the blastocyst is used for stem cell research, embryonic stem cells seem to be more flexible than adult stem cells because they have the potential to produce every cell type in the human body. Scientists can induce embryonic stem cells to replicate themselves in an undifferentiated state for very long periods of time before stimulating them to create specialized cells. There are two sources from where embryonic stem cells can be derived. They are in vitro fertilization and nuclear transfer. Embryonic stem cells are easily grown and are assessed for their fundamental properties. These embryonic stem cells are later grown into the specialized cells and used for different therapeutic and research purposes. However, there has always been an ethical conundrum all over the world within the science community and in public which have strong opinions and has led to controversial discussions regarding the potential of embryonic stem cell research. Thanks.